So, carbohydrates. Cells use carbohydrates as a source of energy and a way of storing energy. The source of energy is usually small, simple sugars such as glucose. Plants can store this in the form of starch, whilst animals store their sugar as glycogen. Carbohydrates are made of three elements: carbon, hydrogen and oxygen. We can organise carbohydrates into different groups based on their size. So what are we going to learn about in this lesson? Well, we're going to learn about the difference between the different types of carbohydrate, as I said, based on their size: monosaccharides, disaccharides and polysaccharides. We're going to look at the structure specifically of glucose and the two types of glucose, which are alpha and beta. And we're also going to look at the structure of ribose. We're going to explain the idea of condensation reactions, which are used to form glycosidic bonds, which you need to turn simple sugars into bigger, longer sugars. And we'll also look at how hydrolysis reactions can break those bonds. And we're going to state the structures and bonding in the following molecules: sucrose, lactose, maltose, starch and glycogen. And we're going to look at how glucose, starch and glycogen and cellulose, structure of them, relate to their function. So we can split carbohydrates into groups based on how big the molecules are. Monosaccharides are single sugars with 1 unit, disaccharides have 2 sugar units, and polysaccharides have more than 2 sugar units in them. Monosaccharides are very, very simple, just one single sugar, as I said. They could be pentose sugars, they're in the shape of a pentagon, they've got 5 carbon atoms. The examples are ribose and deoxyribose, the sugar that you use to make DNA. Or they could be hexose sugars in a hexagon, such as glucose, fructose and galactose, and they all have 6 carbons in them. Now, you need to know the structure of ribose in more detail. Here it is, okay, 5 carbons in a ring, like I said, but you can see you've got an oxygen at the top, and the 5th carbon comes off at the top as a branch, CH2OH. So you've got to try and learn that structure. Okay, and we also need to know about glucose and the 2 types of glucose. All right, now here they are. Here's a picture of them: alpha and beta glucose. They're what we call isomers, okay, because they've got the same formula but a slightly different structure. Now you may be looking at this thinking, well, where is the difference? They look pretty similar, okay? Well, actually, there's only one difference. It's very, very small, and it's on that first carbon. We label the carbons in a clockwise direction, starting on the right hand side after the oxygen. So we've got carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and then carbon 6 off the top. And you'll see on carbon 1, you'll see that the hydrogen and the hydroxide group have swapped over. Now this may seem very, very simple and not a significant change, but it does have a significant change when you start combining these molecules together to make polysaccharides. Okay, so disaccharides, 2 sugars, disaccharides, they're made up of 2 monosaccharides joined together by a bond called a glycosidic bond. Okay, there's 3 examples that you need to know about: maltose, sucrose and lactose. Maltose is made up of 2 alpha glucoses, sucrose is made up of an alpha glucose and a fructose, and lactose is made up of alpha glucose and beta galactose. So how do you take 2 simple individual sugars, 2 monosaccharides, and join them together to make a disaccharide? You've got to do a condensation reaction. Okay, if you watched my video on lipids, you'll know that that condensation reaction is something where you remove water. So we're going to take 2 alpha glucoses in this example here, okay, and you can see that we can remove H2O, and then we can form a new bond there with the oxygen in this condensation reaction, which is called our glycosidic bond. More specifically, this bond here is known as a 1,4 glycosidic bond because it's between the first carbon on the left-hand sugar there and the 4th carbon on the second sugar. So it's a 1,4 glycosidic bond. Now you can do the opposite reaction as well to split a disaccharide into 2 monosaccharides, and this is called a hydrolysis reaction, where you put water in. Now if you continue doing that process and adding on more sugars and doing more condensation reactions, you'll make a polysaccharide. Now these are perfect for storing sugar in a cell because, number 1, they're insoluble, so they're not going to affect the water potential inside the cell, which could cause all sorts of problems with osmosis, and they are compact, so they don't take up very much space. Now you need to learn about starch, glycogen and cellulose in more detail. So let's look at their structures. So starch is the energy store used by plants. Okay? When the plant makes glucose during photosynthesis, it stores it as starch because it's insoluble and it's compact. Now starch is actually made up of two slightly different compounds. One is called amylose and the other one is called amylopectin. Okay? Amylose is made up of about between 200 and 5,000 alpha glucose molecules joined together by 1,4-glycosidic bonds. You just keep joining alpha glucose molecules together. But when you do that, there's a slight angle when you join two alpha glucoses together, and if you keep going, it will eventually start spiraling round. So amylose forms a coil. Amylopectin still has alpha glucoses joined together by 1,4-glycosidic bonds, but it also has some 1,6-glycosidic bonds forming branches coming off, as you can see from the diagram. Now this is useful because those are easy to break off to give quick release bit of energy. Glycogen in animals is very similar to this amylopectin found in the starch. It just has even more branching. And actually glycogen is used for the storage in fungi as well. So again, same reason, because you can break off branches quite quickly to get a quick release of energy. Cellulose, found in plants, you will have come across it before when you learned about plant cell walls. It's very strong substance which stop plant cells from bursting. Now, in order to achieve this strength, they have to have a very specific structure. Now, they're not made out of alpha glucose molecules. Cellulose is actually made out of beta glucose molecules, still joined by 1,4-glycosidic bond. But to avoid this coiling that we had in the amylose, the beta glucose molecules are inverted every other molecule. Okay, so one goes up, the other goes down, up, down. And that will mean that the bond will be regular and you'll end up with a nice straight molecule. And when you get the straight molecule, you can have more straight molecules, okay, and you can line them up and you can join all these straight strands together using hydrogen bonds between them, and that will end up forming fibrils, and those can be bonded together to make fibers, and you end up getting this nice strong material. So just looking at the functions of these polysaccharides in a little bit more detail, starch in plants provides a mixture of quick and slow release energy due to the combination of the amylose, which is quite hard to break down, and the amylopectin, which has the branching, which is easy to snap off. Glycogen in animals is similar to the amylopectin. You can get quick release energy by breaking off those branches, which is useful for respiration. Cellulose is actually very hard to digest, as many organisms do not possess the enzymes required to break these 1,4-glycosidic bonds between the beta glucose molecules. So ruminants, such as cows, actually use bacteria living in their gut to help digest their food.